Hello everyone, welcome to MMBS111 Introductory Microbiology. This is mini lecture 13.4. We're going to be discussing stages 3 and 4 regarding the T cell response and how T cells become specialized and differentiated. There are 5 learning outcomes for this mini lecture. First, after stimulation, name the 3 major types of cells that CD4 T cells will differentiate into. 2, after stimulation, name the 2 major types of cells that CD8 T cells differentiate into. Describe the function of each of the T cell types. 4, what are gamma delta T cells? And finally, what are natural killer cells? So the T cell response is responsible for cell-mediated immunity. T cell reactions are very complex and diverse. They involve a number of different subsets of T cells and a variety of cytokines. Actions of T cells are basically determined by the antigen-presenting cells that activate them, whether it's an MHC1 cell or an MHC2 cell. So T cells activated by an MHC1 antigen-presenting cell will be a CD8 cell, and T cells activated by MHC2 cells will be CD4 cells. All T cells produce a variety of cytokines that contribute to B cell differentiation and inflammatory response. And the end result of stimulating T cells is the activation and mobilization of other T cells, B cells, and phagocytes to the site of infection. This table is from your text, and it shows the types of T cells that differentiate from an activated T cell, and what the receptor is, and then also the function and features of that cell type. So you can see that there are 3 different forms of T helper cells. These are all CD4 cells. And the T helper cells are going to be responsible for assisting in the inflammatory response, proliferating B cells, and activating that cell-mediated pathway. You can also see there CD4 cells also differentiate into the T regulatory cells that control the specific immune response. These cells are really important in preventing autoimmunity and an excessive response from T cells. And then we have our cytotoxic T cells. These are CD8 cells. They lyse foreign cells, and they're really important in the destruction of complex microbes, cancer cells, virus-infected cells. They're also responsible for things like graft and tissue rejections, and they're stimulated, as we saw before, by the MHC1 complex. All right. So as you saw on the chart, there are 3 different types of T helper cells. CD4 is their primary receptor. They are critical in regulating immune reactions to antigens. They also activate macrophages and stimulate the growth of T cells and B cells. And that's what, in the table you notice there are a number of IL numbers. Those are interleukins. They are cytokine compounds that are involved in stimulating the growth of T cells and B cells. So we also have our cytotoxic cells that kill other cells. These cells have CD8 receptors, and they can destroy a number of cells, including those that are infected by viruses. They can attack abnormal self cells like cancer cells. They also attack cells from other animals and humans. So these are the cells that are responsible for organ rejections and issues with tissue grafts. So we have another class of T cells called the gamma delta T cells, and these are distinct from other T cells because they're sort of a T cell hybrid. They have T cell receptors, but they also respond to antigens similar to the way non-specific white blood cells respond, like our neutrophils. So they respond to certain kind of PAMPs, but then they also have T cell receptors that recognize specific antigens. These cells are, because they respond to PAMPs, like our non-specific white blood cells, they're able to respond more quickly than other T cells, and they do produce memory. So once these cells have been activated, when they recognize that antigen, then they are going to proliferate into multiple clones that will then have memory for that antigen. They are particularly responsive to certain types of phospholipids, and they also react to tumor cells. And these cells are considered kind of a bridge between non-specific and specific immune responses. So they kind of have components from both of those response types, which assists in their ability to respond so quickly. We also have some other cells that are considered T cells. We have natural killer cells. These cells are not specific. So while they are T cells, they do not respond to specific antigens. They circulate through the spleen, blood, and lungs. It's hypothesized that they are probably the first killer cells to attack cancer cells and virus-infected cells. And these cells respond and kill other cells in a similar way as our cytotoxic T cells. We also have natural killer T cells. These are a hybrid cell that's a part killer cell, part T cell. It has the T cell receptors for antigens. It's able to release cytokines. So they are also considered one of those bridge molecules or cells between specific and non-specific immunity. All right, so this diagram is also from your text. It can be found on page 362, and this illustrates the activation and then differentiation of your CD4 cells and your CD8 cells. So you can see you have an antigen presenting cell in both situations. One has an MHC2 marker while the other one has an MHC1 marker. So MHC1 is going to activate our CD8 cells, and they will differentiate into our cytotoxic T cells, and then our MHC2 cell is going to then present antigen to our CD4 cell, and those are going to differentiate into our helper cells and our regulatory cells. Okay, so make sure that you are familiar with the types of T cells, what they do, how they are stimulated to specialize. And it might be a good idea to review this presentation more than once. Don't spend too much time on the small details. Really focus on particularly the table on page 360 that distinguishes each specialized type of T cell, whether it was a CD8 or a CD4 T cell, and then its primary functions. Make sure that you're aware of those. All right. That is the end of section 13.5. Here are some of our checking in questions. So make sure you go through these, that you can understand them and answer them correctly. Also remember to go back, review the learning outcomes, be sure that you've mastered those, and if not, remember you can watch this multiple times. I would encourage you to do that, as well as reading the chapter more than one time so that you have a good understanding of these cells. Here's our critical thinking question. HIV is a disease of the immune system where the HIV virus attacks and kills CD4 T cells. When the CD4 blood count is below 500, the individual is said to have AIDS. What immune functions will be compromised as a result of low CD4 cells? So think back to those CD4 cell types. What do they do? What other parts of the immune system are then affected by the action of those cells? And then construct your response. You can find my answer on the next slide, so you can compare your response to mine. And hopefully this video was helpful.